So why do we care about the type of blood that someone has? There's two really important reasons to pay attention to the type of blood. One is mismatched transfusion reactions. So if you're not paying attention to what kind of blood someone has and you give them blood of the wrong type, it can cause their blood to clump up and can kill them. Good reason, huh? The other reason is another thing called hemolytic disease of the newborn. This is where if a mother has a baby of a different blood type than they do, while the baby's in utero, the mother's antibodies can actually attack the baby's blood and cause the baby's blood to clump up. Obviously, we don't want that to happen either. So what really is blood type when we're talking about it? On the surface of a red blood cell are a bunch of different proteins and lipids. In this particular case, we're looking at surface antigens, which is really anything that's on the surface of a red blood cell that could be recognized as foreign by your immune system. So in this particular case, it is a glycolipid. So there is a lipid portion that's stuck in the membrane of the red blood cell, and then it's got some sugars sticking out the end. Type O blood, or the antigen O, there is actually a glycolipid found on the red blood cells. It's not nothing. And that antigen has these 5 sugars in this particular order. In order to get type B blood or type A blood, you have to have an antigen be translated that adds a sugar onto that. So in the case of B type, the B enzyme in red blood cells adds this particular sugar on the end of the glycolipid that's present. And in the type of A blood, a different sugar is added. So it's just one additional sugar on the chain of sugars that's on a particular lipid made and stuck in the membrane of a red blood cell. We can test this in the lab. So there are antibodies that you can buy that the anti-A is actually an antibody that will bind to that A glycolipid and cause the red blood cells to clump up. And what you're seeing is a bunch of red blood cells all clumping up from that antibody when you look at typing blood. In this particular example, anti-A, when anti-A antibody was added to this sample of blood, the A clumped. That means it had to have had the antigen for A on its surface for that antibody to find and cause it to clump. In this particular case, an antibody against type B did not find that B modification on the glycolipid, so it did not cause the blood to clump because the antibody had nothing to bind to clump. And the Rh factor, or anti-D, did find that Rh factor as well and cause the blood to clump. So this person that we're typing their blood has type A positive blood, meaning the A antigen is present, that particular glycolipid modification on the red blood cell, and there's an Rh antigen on the red blood cell, but there is no B antigen. The main types of blood that are present, type A, you remember you've got 2 alleles for every antigen. So in type A blood, this person inherited the enzyme to make that A modification from either one of their parents or both of their parents. And looking at the phenotype, which is type A blood, you don't really know their genotype. You don't know if they're homozygous for the A enzyme or if they're heterozygous and only have one copy of that. Same with type B blood. You know the red blood cells that person makes have the enzyme to modify the B antigen, but you don't know whether they have 2 alleles for that or 1 allele, whether they're homozygous or heterozygous. In the case of type AB blood, you know they had to have gotten the enzyme to modify the A allele from one parent and the enzyme to modify the B allele from another parent. There is no possibility of homozygous. You know they have to be heterozygous and have gotten an A from one, a B from the other. And in the case of type O blood, you know that they did not get an A antigen and they did not get a B antigen, so they can't modify any of them, and they just start with that original glycolipid structure and it never gets modified, so it has to be homozygous. This chart shows two different ways of showing genotype. You may be most familiar with the capital A, capital B, but you can also see different places the I with a B or an A, just different ways to illustrate the genotype. Interestingly enough, for blood, when you are born or after you're born, we already have antibodies in our blood that are ready to attack any A or B antigens that we don't have. So, for example, if I have type O blood, that means I do not have A antigens or B antigens on my red blood cell. That means that I will have antibodies ready to attack A and ready to attack B already present in my blood. It doesn't really make a lot of sense how you could end up with antibodies without exposure to those antigens. So we really have no idea how those antibodies come to be. And it's unusual. Most of the time in our immune system, you have to be presented with something before you develop an immunity against it. But this is true of blood typing. So what that means is, in general, if I have type A blood, I am going to have antibodies floating around in my blood against B, ready to attack B antigens if ever a red blood cell comes into my body that has the B antigen on it. Similarly, if I have type O blood, I don't have any antigens. I have antibodies against A and antibodies against B, ready to attack either one of them. So what that means is any antigens that are not on my red blood cells, I'm ready to attack. So you can already predict who can get donated. So for example, the most difficult one is type O blood. Type O blood has no antigens on the red blood cell surface, neither A nor B. It's just got that original O glycolipid. So it's got, anybody who has type O blood has antibodies ready to attack A and ready to attack B. So they can only receive blood, type O blood. If you give them A blood, if you give them B blood, or if you give them AB blood, their antibodies are going to attack it, clump it up, and have a transfusion reaction. Conversely, if you have type AB blood, you have both A antigens and B antigens, so you don't have antibodies against A or B, and pretty much you could get any blood at all. You're the universal recipient. You can give me A blood, you can give me B blood, it doesn't matter. Interestingly enough, researchers have actually found enzymes that will cut off that extra sugar on the A-type glycolipid or the B-type glycolipid and make all blood O. And there is actually a machine that they're in clinical trials right now. You could buy it, have it in the hospital, and anybody who donates blood, you just stick it in the machine, chop off that extra sugar that gets added on, clean it up, and everybody gets O blood. So maybe there'll be a time in the not so distant future when we don't have to worry about blood typing. All blood that's donated will just be scrubbed and can be given to anybody. It'll be awesome. So there is one other than just the A and B. There is also an Rh factor that we should talk about. In fact, Rh is a little more complicated than A and B in that there are a bunch of different Rh antigens. So there's a whole family of antigens that are found on red blood cells. One in particular, though, tends to cause this hemolytic disease of the newborn, so we pay more attention to it than the other factors that don't cause as much problems. So if you have this particular Rh antigen, you're said to be positive. If you don't have the antigen, you're said to be negative. So here's where the problem happens. If a mother is Rh negative and the father of her child is Rh positive, now Rh is not like A and B. You don't automatically have antibodies against what you don't have. So the mother on her first pregnancy, she's negative, is the only time the woman has problems. If she's positive, there's not any problems. So if she has a child that is positive, now her blood is being exchanged with the baby's blood, and she will develop antibodies against that Rh because she's Rh negative, does not have Rh, but the baby is positive and has Rh. It's not really a problem with the first pregnancy, but those antibodies really have a chance to clonally expand, and by the time she has a second pregnancy where she's positive again, she's got lots of antibodies, and that hemolytic disease of the newborn will happen where her antibodies attack this baby and cause the baby's blood to clump. So in order to stop that from happening, what they'll do if you're an Rh negative mother, they just automatically will give you something called RhoGAM. It's anti-Rh antibodies to try to stop the mother from making antibodies. So every time pretty much a woman does a pre-check into the hospital to have a baby, they'll give her that RhoGAM to make sure that she's not developing antibodies. So really, the amount of people who have type A or type B or type O blood differ in different populations. The most common types are A and O in North America. Pretty rare is AB, pretty much depending, even if you're Caucasian or Asian, it's pretty low, but it depends. So different blood right now is in more demand than other blood, depending on how much of that type blood is needed for donation to people who could be injured.